Welcome to the VBRI. I'm Bill Cashdollar. I direct the core laboratories and the shared research resources at the Institute. Where we're located in the building is our central entrance to the space that actually divides the two halves of the building. To my left, the west side of the building was the first part of the BRI established in 1991. To my right and to the east, that portion of the building was added on in 2006. If you look straight forward to the south, we are putting an addition on this space that will almost double the size of our research capacity at the BRI when it's eventually completed. So if we move this direction, a couple features that I want to point out as we go to the east side of the building. On this wall to my left is a history of innovation at the BRI, and it highlights a couple things that I think are important to keep in mind. Back in the 40s, the founders, the Junior League of Wisconsin, always had it in their mind that not only did they want to provide blood products to the community, but they also wanted to do research surrounding transfusion medicine and blood health. And you can see on this wall some from highlight discoveries over the years. On my right here, what you'll see are some images of chairs that exemplify donations that have been provided to support the research of established investigators at the BRI. Gives them a little bit of flexibility to be creative and push the boundaries of their science. So if you move this direction, to my left is a large conference room that holds about 110 people or so, where we have our seminars. We have invited speakers all the time, bringing discoveries, presenting them to our scientists so that it increases our knowledge base and collaboration. Go up to the second floor. So this moves into the laboratory space on the east side of the building. Gives you a feel for what the labs look like on this side and how they're arranged and how they meet the needs of the scientists. So this is a typical laboratory module on the east side of the BRI. This module is designed to hold 4 scientists in support of a given research program. The lab members can be graduate students, postdoctoral fellows, scientists, technologists, who are working on a given research area. This particular scientist, he studies natural killer cells. There are cells that circulate throughout the body that are designed to recognize and kill virus-infected cells and tumor cells. And what Millar's lab is trying to do is to make those natural killer cells better killers of cancer so that they can recognize tumors and eliminate them from the body. 2 things you can see from this particular lab where we're beginning to put the foundations of the building in place. And then to my left is the tower crane. Our tower crane has to be very tall so it can move into a very tight space, move heavy materials in a tight space. And we have in this area of the building some high-end microscopes that are specifically targeted for various applications. And the one we'll look at now allows us to look at blood flow in a live animal model of various disease states in real time. And so what we can do is we can do an animal surgery, expose a vessel, and then injure that vessel to mimic what happens when you cut yourself. This first video just shows blood flow. So you can see how fast that blood flows, and you can see the discoid shape of the red cells here. This picture illustrates blood flow from the top to the bottom, and the platelets in this system are labeled green. And an injury was made right there where I'm showing that my arrow is pointing, and you can see platelets accumulating, and they're important in blood clotting. Platelets are accumulating at the site of the injury, and that's all normal. This animal model is not a normal situation. You can see how much larger the platelet accumulation is, and then these proteins that appear in red, that's fibrin. And in this particular animal had a defect in its ability to scavenge fat, and so this is not normal, and this is what can happen in somebody who has severe atherosclerosis, and that's what this model is designed to study. All right, so we're moving down to the north side of the building now. This is a breakout space where scientists can come and just sort of relax, have a cup of coffee, eat their lunch. Research is time-consuming, intensive, and can be sometimes frustrating. And so sometimes it's just good to get a break. As we move into this space, on either side of the hallway are two more of the shared resources that I direct. To my right is a facility where we make viral vectors. I personally am a virologist by training. On the left side is the molecular biology core. So we have a DNA sequencer, we have a robot to help us set up libraries for DNA sequencing. As we walk over, I could give you a little bit more of a history of myself. I started on this campus back in 1984 as a faculty member at the Medical College of Wisconsin. I've maintained that faculty appointment. Almost all of the faculty here at the BRI have faculty appointments at the medical college. We are an academic institution. This is our admin space that continues where investigators have their offices on either side of the hall, and in the central area are administrative support functionalities. If you look on the wall here, there are various covers of journals where our investigators publish their work. And if one of our investigators has a particularly impactful article and they have a cool graphic that might help illustrate their research, sometimes they put those graphics on the cover. If you're an athlete, you're on the cover of Sports Illustrated, or if you're a musician, on the cover of Rolling Stone. So I think we're very proud of the fact that our investigators have published in these journals. So we're back again to the midpoint of the building, and here is the transition to the west side where this was, again, the building was finished in 91. You can feel there's very little demarcation between the east and the west side in the admin space. It feels very continuous, and the hope is when we put the new addition on, that centralized entrance will still be the hub of activity between all three portions of our space. We want it to be collaborative and interactive. That's important. So we're moving now into the west side of the building, and these laboratories are set up a bit differently. They feel more like an individual room. The laboratories that are assigned to the principal investigators are around the outside of a big square, and the internal space is devoted to shared resources and other support functionalities. This laboratory is headed by Christian Castro. He's an investigator that studies bleeding and in very catastrophic situations like battlefield wounds and car accidents, where an individual who's injured needs to be stabilized quickly. So let's move on to another one of the shared resources. This happens to be one of our busiest, and that's the flow cytometry core. This laboratory has several instruments in it that are used to analyze cells. These two instruments will do as many as like 12 to 18 colors, basically fluorescent tags, to analyze a particular cell population to determine a particular cell type. That instrument can do as many as 40 parameters. And so that power of identifying a particular cell type is used by all the investigators in the building in one application or another. So this is a very busy operation normally. And all this instrumentation in this central facility has applications that various of the labs use at one time or another. We currently have 29 bench scientists in the building, and we plan to expand to 40 to 45 eventually, depending on the size of their labs, when we have additional space. And so that'll allow us to have a critical mass in the different areas of research that we focus on. All of it's blood research. That's a commonality. Science is becoming more of a team sport, if you will. The days of the individual lab that runs all by itself without any interaction with other people is pretty much gone. People work together now. They have to. The problems are hard, and they need to work with each other to accomplish their research goals. We are always open to showing people around. Some folks consider us, the BRI, as maybe one of the best kept secrets in the Milwaukee area. We don't want to be the best kept secret. We want people to know about what we do. We're proud of what we do, and we want to reach out and have people know how important the work is that we do here. That's all I got.